We will cover the definition of adaptive immunity and classify the T cells according to its types, discuss the functions of CD4 and CD8 T cells with respect to the activation, its co-stimulation and memory function. And the last one is to discuss the effects of the superantigen on T cells. So what is immunity? The main function of the immune system is to prevent or to limit the infection. Or immunity can be defined as a resistance of a host to pathogens and their toxic effects. So we can divide the immunity into innate and the adaptive immunity. The innate immunity relies on the mechanism that exists before infection, and it functions immediately, but it's not specific. So the second one is the adaptive immunity. It relies on the mechanism that adapt after infection. And in adaptive immunity, the T and the B lymphocytes are involved. And the one cell determines the one antigenic determinant at a time, and it is highly specific as compared to the innate immunity. So innate immunity relies on a preformed components, and that's why its response is rapid within minutes of the infection. As already told that it is not specific and it has no memory cells, and the same response after the repeated exposure, and does not lead to the clonal expansion of the cells as compared to the adaptive or the acquired immunity. So in adaptive immunity, it is based upon the resistance acquired during life. Adaptive immunity occurs after exposure to an agent, and it becomes stronger upon repeated exposures, and it is specific. That is, each cell responds to a single epitope on an antigen. The adaptive immunity has anamnestic memory, that is, its repeated exposure leads to faster, stronger response, and it leads to the clonal expansion of the cells. So adaptive or acquired immunity may be active or passive. So in active immunity, it occurs by a natural exposure to an infectious agent in which the antigens naturally enter in body, or by immunization, in which a weakened or dead antigens are introduced to the body and the body produces antibodies for the antigen. While in passive immunity is transferring antibodies from an individual who is immune to a certain antigen to someone who is not. For example, the antibodies pass from mother to fetus during pregnancy, that is immunoglobulin G, and in breast milk, that is immunoglobulin A. So another example is antivenom injections to treat the snake bites have antibodies. So this is the active and the passive immunity. Now the adaptive immunity has two components, that is the humoral immune response and the cell-mediated immune response. So first we will discuss the cell-mediated immune response of the adaptive immunity. So in this lecture we will discuss the cell-mediated immune response, which is provided by the T lymphocytes, and this cell-mediated immunity can recognize only protein antigens, and it recognizes antigens presented by the antigen presenting cells attached with the class 1 or class 2 major histocompatibility molecule. And this cell-mediated immunity provides immunity to intracellular bacteria, viruses, fungi, and protozoa. and it causes type 4 hypersensitivity and causes acute graft rejection. So the cell-mediated, now we discuss the origin of the T and the B cells. So this picture shows the origin of the T and the B cells. Stem cells are the precursor of both T and B lymphocytes and resides in the bone marrow. These stem cells will differentiate into T cells in the thymus, either CD4 positive helper T cells or CD8 positive cytotoxic T cells. So the T cells differentiate into the thymus, into the CD8 and the CD4 positive helper T cells. On the other hand, the B cells differentiate in the bone marrow, which later on differentiate into the plasma cells, which produces large amount of antibodies. So the stem cells resides in the bone marrow, and then the T cells differentiate into the cytotoxic and the helper T cells in the thymus, while the stem cells of the B cells differentiate into the plasma cells, and which secretes the antibodies. Now the thymus as a site for the positive and negative selection. When the T cells enter in the thymus, they lack antigen receptors and the CD3 and CD4 and CD8 proteins on their surface. Now this picture shows the section of the thymus showing the subcapsular region, cortex, and this is a corticomedullary junction, and this is the medulla. So when the T cells enter the thymus, they lack the antigen receptors and the CD3, 4 and the 8 proteins on the surface. Means they are double negative. That means they don't have a CD4, they don't have a CD8. They are double negative thymocytes. What happens? That first they differentiate into the T cells that express both antigenic receptors and the various CD proteins on the surface. So first the stem cells were double negative, CD4 nor CD8. Then they express both CD4 and CD8 and become double positive. And then they are going to mature and express either CD4 or CD8. What happens when these double positive cells, if it comes into contact with these double positive cells, if it comes into contact with a cell bearing class 2 MHC protein, it becomes CD4 positive. Or if these double positive comes into contact with cell bearing class 1 MHC protein, then it will differentiate into CD8 positive cells. So these double positive cells and double negative cells located in the cortex, while the single positive cells located in the medulla. So the thymus is a site where positive and negative selection occur. What is positive and negative selection? We will discuss later. We'll discuss in the later slides. So positive and negative selection of the T cells. Within the thymus education occurs. It has two processes. First one is the clonal deletion. What is clonal deletion? That is, those CD4 and CD8 positive cells having antigen receptors for self proteins are killed by the process of apoptosis, and this is called the clonal deletion. And the removal of these self-reactive cells is called negative selection. This negative selection results in tolerance to our own protein, that is self-tolerance, and prevents autoimmune reaction. So this negative selection results in a self-tolerance to some extent. The second process is a positive selection. What is positive selection? In positive selection, it is those CD4 and CD8 positive cells having antigen receptors that do not react with the self-MHC proteins are also killed. It means positive selection results in selection of T cells that react well with self-MHC proteins. So the positive selection results in self-MHC restriction and the negative selection results in a self-tolerance. So this is the thymus education. The thymus education have two processes, that is a positive selection and a negative selection. So the T cell progenitors develop in the bone marrow and migrate to the thymus. Then there is a positive and negative selection occur in the thymus. Then the mature T cells migrate to the peripheral lymphoid organs, and then these activated T cells migrate to the sites of infection. Now comes the classification of the T cells. The lymphocytes differentiate into the T and the B lymphocytes. Then the T cell progenitors differentiate under the influence of the thymic hormones into T cell subpopulation. These cells have surface glycoproteins called CD4 and CD8, and all T cells have CD3 proteins and antigen receptor on their surface, that is T cell receptor. So the T cells are divided into the two main classes on the basis of CD4 or CD8 proteins on their surfaces, as discussed earlier. So the mature T cells have either CD4 or CD8 proteins, but not both. So the CD4 lymphocytes, also called the helper T cells, has three main functions. They help B cells develop into antibody producing plasma cells. They help CD8 T cells to become activated cytotoxic T cells. They help macrophages to kill ingested microbes. And all these functions are performed by three subtypes of CD4 cells, that is Th1, Th2 cells and Th17 cells. So the Th1 cells produces gamma interferon that activates the macrophages and the cytotoxic T cells. The Th2 cells helps to activate the B cells activation and the differentiation of B cells by producing interleukin 4 and the interleukin 5 that enhance the growth of and differentiation of the B cells into the plasma cells. And the Th17 cells produce interleukin 17 that recruits the neutrophils to the site of inflammation. So the Th1 cells stimulates the cell-mediated immunity, while the Th2 cells mediates the humoral immunity, because the Th2 cells involve in the differentiation and the growth of the B cells, while the Th1 cells activates the cytotoxic T cells. That's why the Th1 cells involve in the cell-mediated response and the Th2 cells stimulate the humoral immunity. Now the cytotoxic T cell, that is the CD8 positive T cells, performs cytotoxic functions. That is, it is involved in killing the virus infected tumor and allograft cells by releasing the perforins or by the apoptosis. So this is all about the CD4 and the CD8 T cells. Now come to the activation of the T cells, how the T cells becomes activated. Now, before discussing the activation of the T cells, we should know that what are the antigen presenting cells. We should know that the dendritic cells, macrophages and the B cells are the most important antigen presenting cells. Now come to the activation of the T cells. First, we will discuss the activation of helper T cells, how the helper T cells becomes activated. It begins when the antigen presenting cell engulfs the microbe, and within the cytoplasm, the microbe breaks down into the small fragments or peptides. These fragments and peptides attach with the class 2 MHC protein. And then this complex of MHC2 and peptide transported to the surface of antigen presenting cells. So this complex is recognized by the T cell receptor on the helper T cell or the CD4 positive T cell and attached to it and become activated. So this is the process of helper T cell activation. Now come to the activation of the cytotoxic T cell. In this, the antigen presenting cell itself is infected with a virus, and within it viral proteins are prepared and then present on the surface of antigen presenting cell in association with the class 1 MHC protein. Then this complex of MHC1 and viral protein recognized by the T cell receptor of cytotoxic T cell and attached and becomes activated. So this is the process of the activation of the cytotoxic T cell. Now in this picture on the left side, we can see the activation of the helper T cells in which the antigen presenting cells processed antigen in association with the class 2 MHC protein, and the antigen is recognized by the T cell receptor of the helper T cell and the antigen is recognized by the helper T cell receptor, and the helper T cell becomes activated to produce the interleukin 2, which binds to its receptor on the helper T cell and further activate it. So on the right side we can see that a virus infected cell presents a viral antigen in association with the class 1 MHC protein. Then the viral antigen is recognized by, so this complex of viral antigen and the class 1 MHC protein is recognized by the T cell receptor of the cytotoxic T cell, and it is attached and activated and kill the virus infected cell. So again, I hope that the activation of the T cells, activation of the helper and the activation of cytotoxic T cells will be clear. So remember the rule of the 8. What is rule of the 8? That it's the MHC1 always attached to the CD8 T cells and the MHC2 attached with the CD4 T cells. This is the rule of 8. So now comes to the co-stimulation to activate the T cells. So the two signals are required to activate both the helper and the cytotoxic T cells. First signal we already discussed, that is the interaction of the antigen and the MHC protein with the T cell receptor on the helper and the cytotoxic T cells. And the second is the co-stimulatory signal. What is co-stimulatory signal? Co-stimulatory signal is the interaction of the B7 protein on the antigen presenting cell with the CD28 protein on the helper T cell, or same as in the case of the cytotoxic T cell. So this co-stimulatory signal is important because interleukin 2 is made by the helper T cell, and this interleukin 2 make the helper and the cytotoxic T cells capable of performing its regulatory, effector and memory functions. That's why the co-stimulation to activate the T cells is very important, because it is involved in its regulatory, effector and the memory function. So what is memory cells? The memory cells live for the many years or have the capacity to reproduce them. A large number of memory cells are produced, and so the secondary response is enhanced and is greater than the primary response. Basically, the memory T and the B cells have the ability to respond rapidly and vigorously for many years after the initial response to a microbe or other foreign material. So the memory cells, so this memory response to a specific antigen is due to several features. Many memory cells are produced so that the secondary response is greater than the primary response. Memory cells live for many years or have the capacity to reproduce themselves. Memory cells are activated by a smaller amount of antigen and require less co-stimulation than do the unactivated T cells. And the activated memory cells produce greater amount of interleukin than do the unactivated T cells when they are first activated. So this is all about the memory cells, memory T cells or memory B cells. Now come to the effect of the superantigen on the T cells. What is superantigens? Now the superantigens are those antigens that can activate large number of T cells without internal processing, as we discussed in the previous slides. For example, for the internal processing of the virus and the antigen. So these superantigens may be a staphylococcal enterotoxins or maybe a toxic shock syndrome toxins. So these both toxins are the example of the superantigens that can activate the large number of the T cells without internal processing. So in the top of the picture we can see that the helper T cells is activated by the presentation of processed antigen in association with class 2 MHC protein. So this is an antigen and this is the class 2 MHC protein, and this complex is attached to the T cell receptor and activate the helper T cell as we discussed earlier. But in case of the superantigen, what happens that in the bottom picture, the helper T cells is activated by the binding of the superantigen, and this superantigen is attached outside of its antigen binding sites, as we see in this picture, and this superantigen is attached without being processed by the antigen presenting cell. And as I said earlier, the superantigens can activate the large number of T cells without internal processing. So in this lecture, we discussed the definition of the adaptive immunity, and we discussed the origin of the T and the B cells, and thymus for a site of a positive and negative selection, the thymus education, including positive and negative selection, and types of T cells, CD4, CD8, and further subtypes of the CD4 lymphocytes involving Th1, Th2 and Th17, and what are the main functions of the Th1 versus Th2 cells, what are the functions of the cytotoxic T cells, and how the helper and the cytotoxic T cells activated, and the rule of 8, and what is the co-stimulation, and what is the importance of the co-stimulation to activate T cells, what are memory cells and what its significance, and what are the super antigen and how it is stimulated helper T cells. So this is all about the adaptive, cellular arm of the adaptive immunity. In the next section, we will discuss the humoral arm of the adaptive immunity.